Beth Shapiro, How to Clone a Mammoth, The Science of De-Extinction. Ever wondered if science could truly bring back extinct species? Get ready to explore the fascinating world of de-extinction in the summary of How to Clone a Mammoth, The Science of De-Extinction by Beth Shapiro. Discover the motivations behind these groundbreaking experiments and learn about the researchers pursuing the incredible goal of reconstructing extinct animals. Dive into the challenges they face in recovering ancient DNA, the methods they employ, and the ethical considerations involved throughout the process. Also, gain valuable insight into the prospects of reintroducing these revived species into their respective ecosystems, and the potential impact on the environment.
Reimagining extinction. In the past, extinction has been a natural cycle of birth and death, with only 1% of all the animals that ever lived existing today. However, some scientists are looking at de-extinction as a way to restore interactions between species and preserve biodiversity. Although de-extinction is mostly theoretical, researchers have been able to clone sheep and grow embryos of extinct animals. The goal of this technology is not to bring back the past but rather to build a better planetary future by addressing environmental challenges and the loss of potential benefits like disease remedies. De-extinction could be a way to repair the damage caused by humans, who have overhunted and cleared habitats. The resurrection of ecological interactions resulting from de-extinction technology could hold immense value. The Promise of De-extinction. The pursuit of de-extinction seeks to restore ecosystems by reviving extinct species. In 2013, a TEDx presentation at National Geographic discussed the moral, ethical, and research challenges of this technology. Despite the unknown consequences and concerns, scientists are motivated by the potential knowledge to be gained from these endeavors. Ecology students' priorities. Ecology students align with the public's beliefs in restoring extinct animals, preferring herbivores over carnivores and bigger animals over smaller ones. However, practicalities such as evaluating the species' role in a critical ecosystem remain a challenge for researchers. The Challenges of De-extinction. The retrieval of ancient DNA for de-extinction involves extracting usable genetic material, which is a challenging process. A 17-million-year-old piece of amber containing small bees was used by scientists, who had to enter an ultra-clean lab to avoid contamination. Though the results were disappointing, this case highlights the difficulties inherent in this process due to genetic material deterioration and microbial contamination. Revolutionary DNA replication. Scientists have made significant advancements through the polymerase chain reaction, PCR. By duplicating small amounts of a DNA sequence, the PCR method permits researchers to produce millions of copies for experimentation. However, DNA doesn't survive millions of years. Often, animal bones and tissues preserved in cool environments, caves, and dry locations yield the best samples. Accurately dating a sample is crucial, and radiocarbon dating is used to date biological specimens up to 40,000 years old by tracking the decay of a carbon isotope. Reviving the mammoth. Scientists hope to clone the mammoth using preserved genomes found in mummified remains. Volcanic ash can provide a time stamp for fossils, allowing researchers to estimate age and extract genetic material. Currently, no extinct species remains contain somatic cells, which are necessary for cloning through somatic cell nuclear transfer. Researchers hope to find a well-preserved specimen, ideally a mammoth mummy, with unique adaptations to its habitat in closely related living relatives to make de-extinction feasible. However, the cloning of a mammoth remains unlikely in the near future. Despite this, scientists continue to search for more samples, hoping to find the perfect candidate for de-extinction. De-extinction. Two promising paths. Selective breeding and genetic engineering are the most promising options for de-extinction, according to the research. Selective breeding is an ancient practice that uses observable traits to perpetuate desirable characteristics in animals. DNA carries these traits down through generations. However, this method is ineffective with animals that have few offspring, such as elephants. Genetics engineering is a more recent technique that offers possibilities without the need to wait for breeding results. Genetics modification can entail the use of CRISPR-Cas9, a revolutionary technique used to remove or add specific gene sequences. While this technology holds the potential to bring extinct animals back to life, legal and ethical questions need to be addressed. Additionally, the potential ripple effects could lead to unforeseen consequences. Finally, this research emphasizes the importance of sustaining endangered species today and preserving biodiversity for future generations. Bringing back the mammoth. Harvard's George Church and his team plan to de-extinct the mammoth by editing the genome of an Asian elephant. They will compare the genomes of the two species to locate the differences and synthesize mammoth DNA. However, the first revived mammoth will face challenges as its behaviors would need to be learned. One step in the process includes cold-proofing elephant blood, as mammoth blood hemoglobin carries oxygen at low temperatures. While the process is doable, it lacks funding, and scientists must work with limited lab space and resources. Environmental factors can also impact the success of revival. Genetic engineering and reproduction. A thought-provoking exploration of the challenges that come with genetic engineering and reproduction, from ethical considerations of harvesting eggs from endangered species to the use of artificial wombs in supportive environments. The baby animals born this way would still require a place to live and learn how to survive, and scientists are uncertain about whether their natural instincts could be replicated in captivity. Still, the possibilities of genetic manipulation in reproduction continue to be an exciting arena for scientific research. Restoring ecosystems with de-extinction. De-extinction offers technical achievements that can help revive extinct species and restore degraded ecosystems. Plants feed animals, which further support the food system. Among ecosystems, grasslands have suffered the most damage, making restoration to the late Pleistocene era challenging. However, scientists can reintroduce essential animals to attain partial restoration. This motivates rewilding, the return of feral animals to their habitats. Through de-extinction, the means to revive extinct species can further restore the Earth's biodiversity. The role of megafauna in rewilding efforts. Rewilding efforts are likely to focus on big herbivores, such as mammoths, horses, and camels, that can nourish the soil and support carnivores like lions, panthers, and cheetahs. These herbivores are keystone species that play a vital role in the food chain. The challenges of de-extinction. De-extinction faces regulatory and ethical challenges, with concerns ranging from competition for resources to animal exploitation and unknown diseases. Critics, ranchers, and farmers may oppose the introduction of genetically adapted animals, likely leading to policy debates and complex regulations. In addition, de-extinction could divert resources away from existing conservation methods and endangered species protection. While advocates see de-extinction as a way to revive extinct species and restore ecological balance, its success will depend on balancing conservation efforts with ethical and practical considerations. Bringing back extinct species. Advances in science have made it possible for extinct species to be brought back to life through a process called de-extinction. Although the road to success may be long and difficult, the idea of de-extinction symbolizes the power of science to restore the past and create a better future. In the future, children may even get to encounter a woolly mammoth with its luxurious coat, thanks to de-extinction and the wonders of modern technology.
After delving into the captivating theories and developments discussed in How to Clone a Mammoth, the Science of De-Extinction, it is apparent that bringing back extinct species presents immense challenges in the extraction and analysis of their ancient DNA. Although researchers have achieved significant progress in various subfields, de-extinction still has a long way to go before becoming a reality. Furthermore, debates on moral, ethical, and economic concerns continue to shape the prospects of these groundbreaking experiments. Ultimately, the exploration of de-extinction represents the power of science in understanding the past and improving the future, enabling us to ponder the potential of restoring lost biodiversity and building a better planetary future.